Imagine you run this highly secure state-of-the-art factory. The sprawling manufacturing floor, let's call it the cytoplasm, is physically separated from the highly restricted blueprint room.
Right, the nucleus.
Exactly, the nucleus. And the only way to move information from the blueprint room out to the factory floor is through a massive wall fitted with these tiny, rigid, revolving doors.
Which is fine for standard stuff.
Yeah, normally it's totally fine. These doors are about 60 nanometers wide. It works perfectly for everyday blueprints. But what happens when you need to export a single massive blueprint that is literally thousands of times larger than the door itself?
You've got a problem.
A huge problem. You can't dismantle the blueprint because then the factory can't read it. And you obviously can't break the door without, compromising the whole facility.
It is a massive logistical nightmare. I mean, if you force the blueprint through, you completely destroy the genetic cargo.
Right.
But if you don't export it at all, the factory just shuts down. And in a biological system, a factory shutting down usually means the cell dies.
And this isn't just a fun thought experiment for us to kick around today. This is a very real, incredibly complex biological physics puzzle happening inside your body right this second.
Yeah, in your muscles specifically.
Today, our deep dive is focusing on a 2026 paper published in the journal Cell by Zaganelli and colleagues. It's titled, "Nuclear Envelope Budding Enables Export of Large Transcripts in Muscle Cells."
It's such a great paper.
It really is. Our mission today is to explore exactly how your muscle cells solve this impossible shipping problem by constructing a secret, non-standard exit out of the nucleus. This paper completely flips the textbook understanding of how cells move material around. So, okay, let's unpack this.
To really appreciate the sheer elegance of this secret exit, we first have to understand why the normal canonical exit is just completely inadequate for certain types of cells.
Right, the revolving door.
Exactly. In a standard cellular shipping route, molecules leave the nucleus through a structure we call the nuclear pore complex, or NPC. Think of the NPC as that rigid revolving door you mentioned in your factory analogy.
So it's heavily guarded, right?
It's a highly structured, heavily guarded channel. And even when it is fully stretched to its absolute physical limit, it maxes out at about 60 nanometers in diameter.
Which, to be fair, is incredibly tiny, even on a cellular scale.
Oh, it is. But for most of the cells in your body, that 60 nanometer limit is totally fine. Most messenger RNA, or mRNA, which are the genetic blueprints for building proteins, can spool through that pore without a problem.
Because they're small and flexible.
Right, they just make the trip easily. But muscle cells face a highly unique physical complication. During muscle development, which is a process called myogenesis, single muscle cells fuse together to form these long post-mitotic tubes.
And those tubes actually have to function.
Yeah. To contract and actually work, these tubes have to build sarcomeres, which are the fundamental contracting engines of your muscles.
Right. And building sarcomeres isn't like building a standard cellular widget. It requires constructing some of the largest, most complex proteins in the entire mammalian body.
Exactly. And the undisputed king of them all is a protein called titin. The mRNA transcript required to build titin is an absolute monster of a molecule.
It's huge.
It is over 100,000 nucleotides long in its mature, fully processed form.
To give you a visual for just how absurdly big that is, if a normal mRNA transcript is just a regular person walking through that 60 nanometer revolving door, the titin mRNA is a fully assembled commercial cruise ship.
That's a perfect way to look at it.
It simply cannot fit through the nuclear pore complex. And like we said with the factory, if you dismantle the genetic instructions, you can't build the muscle.
It's a profound physical constraint. If a cell were to rely on the traditional textbook model of nuclear export, a ribonucleoprotein complex the size of titin would require such severe structural remodeling that it would likely never make it out intact.
So what does this all mean? If the cruise ship transcript can't fit through the normal door, what is the cell actually doing? How does the cell not just grind to a halt?
Well, that is the exact driving question Zaganelli and the research team set out to answer. And the answer is that the cell completely bypasses the revolving door.
It just ignores it.
Yep. It constructs a profoundly different kind of exit strategy.
So if I'm tracking with you, we've got this giant cruise ship of an mRNA, a door that's way too small, and the inevitable conclusion that there must be a custom hidden door somewhere in the blueprint room. So just a quick recap for everyone. Normal door, the NPC, is 60 nanometers. Titin mRNA is way too big. Result, we need a new door.
Yes, and the researchers discovered this hidden door by looking at a process called nuclear envelope budding, or NEB.
Nuclear envelope budding, okay.
To see this, they utilized transmission electron microscopy, or TEM. TEM essentially fires a beam of electrons through a sample to create incredibly high-resolution images of the internal structures of a cell.
So they can really see the architecture.
Exactly. And when they looked at the undifferentiated muscle cells, the ones not yet building giant proteins, the nucleus looked totally standard. Smooth borders, normal nuclear pores doing their normal job.
Just a regular wall.
Right. But when they looked at the differentiated cells, the ones actively building massive muscle proteins, the inner wall of the nucleus was doing something wild.
The internuclear membrane was actually bulging inward, right? Like stretching right into the center of the nucleus itself.
Yes. It creates these massive inward-facing bubbles, some of them up to a full micrometer wide.
Wow. A full micrometer.
And when the researchers zoomed in even closer on these giant buds, they found smaller bubbles nested inside them. They named these internal luminal vesicles, or ILVs, and these nested bubbles average about 150 nanometers wide.
Okay, picture the rigid, highly secure wall of the nucleus suddenly softening. It stretches out, reaching deep into the blueprint room to swallow that giant cruise ship-sized mRNA whole, wrapping it up in a protective, flexible bubble.
Yeah.
It essentially creates a temporary, custom-built dry dock just for the oversized cargo.
That's a fantastic way to visualize it. But the researchers couldn't just assume what they were seeing under the electron microscope was a transport bud.
Right, it could have been a mistake.
Exactly. They had to prove they were actually looking at the inner nuclear membrane stretching inward and not just some random cellular debris or like a structural artifact. So they used fluorescence microscopy to tag specific membrane proteins.
And what did that show?
They found that these giant inward bubbles contained all the classic chemical markers of the inner membrane, but they completely lacked the proteins found on the outer membrane.
Oh, interesting.
Crucially, they also lacked the rigid, mesh-like lamina coat that usually lines the inside of the nucleus.
Which makes sense, right? I mean, you'd have to dissolve that rigid scaffolding if you wanted the wall to blow a bubble. But how did they know for sure that the giant Titan blueprints were actually inside those specific bubbles?
They used a brilliant, highly specific technique called single-molecule FH, which stands for fluorescence in situ hybridization.
That sounds intense.
It sounds complicated, yeah, but it essentially allows you to deploy glowing fluorescent probes that bind only to the specific RNA molecules you want to track.
So they just lit up the cargo.
Exactly. They deployed probes engineered specifically for the massive Titin transcript and another giant muscle transcript called nebulin, and the results were stunning.
They were inside.
These giant transcripts were highly enriched entirely inside these nuclear buds. Sometimes both Titin and nebulin were packaged inside the very same bubble.
Wait, let me guess. It was only giant transcripts in those bubbles, right? The cell wasn't just throwing everything in there willy-nilly.
That's the crucial detail. They tagged smaller, normal-sized muscle transcripts and standard everyday housekeeping genes that the cell uses for basic survival.
Pen.
Those smaller transcripts completely ignored the custom dry dock. They were entirely absent from the buds, presumably just using the normal revolving doors.
That is so efficient.
Furthermore, the probe showed that the giant transcripts inside the buds were fully processed.
Meaning they were mature, edited, and totally ready to be translated into protein the second they hit the factory floor.
Precisely. And you know what's fascinating here is that this specific biological trick, nuclear envelope budding, isn't entirely new to science.
Wait, really?
Yeah, if we connect this to the bigger picture, the history here is incredible. Biologists have actually seen structures resembling these buds since the 1950s.
Since the 50s.
But for decades, they were almost exclusively associated with viral infections.
Viruses. How do viruses fit into a muscle cell building a protein?
Well, think about herpes viruses. When a herpes virus infects a cell, it replicates its DNA inside the host's nucleus. It then starts assembling viral capsids, the protein shells that hold the virus.
Right.
But those capsids are simply too large to fit through the normal nuclear pore.
Ah. So the herpes virus needs to get a cruise ship out of the nucleus too.
Precisely. The virus triggers the host's inner nuclear membrane to dissolve its rigid lamina, bud inward, swallow the giant viral capsid, and transport it out into the cytoplasm. For a long time, virologists and cell biologists thought this was just a devious, highly specialized trick invented by the virus to escape. But this paper firmly proves that the virus didn't invent the mechanism at all.
It just hijacked it.
It essentially hijacked a normal, ancient, naturally occurring cellular pathway that our muscle cells use every day to build our bodies.
The virus just stole the master blueprints for the custom dry dock. That is mind-blowing. Okay, let's do another quick checkpoint. So mega mRNAs, like titin, are routed to custom bubbles called buds. These buds contain smaller vesicles, the ILVs holding the mega mRNA, and it's a pathway viruses love to steal.
Spot on.
But that realization brings up a massive logistical problem. If the cell is running normal revolving doors for the small stuff and this giant viral-friendly dry dock for the huge stuff, how does it not mix them up?
That's the million-dollar question.
I mean, you definitely don't want a regular-sized blueprint taking up space in the custom dry dock, and you really don't want a cruise ship trying to ram through a revolving door. There has to be some kind of cargo selector making those decisions.
Yeah. You're exactly right. There must be a selector. And to find it, the researchers utilized a relatively new and ingenious technique called proximity proteomics.
Proximity proteomics.
Okay, they took a protein known to sit stably inside these nuclear buds, called LAP1B, and they genetically fused it to an engineered enzyme called turboid.
Curboid.
Now, turboid is a promiscuous biotin ligase, which means...
Wait, hold on. A promiscuous... What? Let me see if I can translate that.
Yeah.
If you fuse this turboid thing to the bud, does it act sort of like a molecular paintball gun?
A paintball gun.
Yeah. Like if you feed the cells the right chemical ammunition, this enzyme just frantically shoots a chemical tag onto any protein that gets close to the bud.
Yes, that is a perfect analogy. You feed the cells a vitamin called biotin, and the turboid enzyme basically paintballs any protein that comes within a tiny 10 nanometer radius of the nuclear bud.
It just tags them.
It permanently splatters them with a biotin tag. The researchers then essentially washed the cells, collected all the proteins that had paint on them, and ran them through a mass spectrometer to see who exactly was hanging around the dry dock.
Okay, so who got caught covered in paint?
A protein called UIF, which stands for UAP56 interacting factor. It's a known RNA-binding protein.
So that's our guy?
Well, finding it near the bud is one thing, but proving it actually controls the cargo is another entirely. So the researchers used small interfering RNA to temporarily silence the gene that makes UIF.
Oh, wow.
They basically fired the cargo handler and removed him from the factory entirely.
Let me guess the outcome. If you delete the only guy who knows how to load the cruise ships, the whole factory backs up, right?
Yeah.
The Titan blueprints must have just piled up in the blueprint room.
The whole system completely backed up. Without the UIF protein present, the massive Titan mRNA entirely stopped going into the buds.
Just stuck.
When they measured it, they found a massive accumulation of Titan mRNA trapped uselessly in the main nuclei of the depleted cells. Meanwhile, the smaller canonical transcripts exported just fine through the revolving doors.
So it only affected the big stuff.
Exactly.
Yeah.
This elegantly proved that UIF is the specific, dedicated cargo handler for the giant transcripts.
Here's where it gets really interesting. I'm trying to visualize this process in real time. We know UIF handles the cargo, and we know the membrane bulges inward to make a bud.
Right.
But the researchers wanted to know exactly how UIF and the mRNA were sitting inside that bud, right?
Yeah.
Like, are they just floating loosely in an open cave on the edge of the nucleus, or are they sealed tight in a bubble?
Yes, resolving that structural question was vital. And to answer it, they performed a beautiful live imaging experiment called fluorescence recovery after photobleaching, or
FRP. Love that acronym.
They genetically engineered the living muscle cells so that the UIF protein would glow bright green under a specialized laser microscope.
Okay, picture this. You're looking through the microscope directly into a living cell's nucleus, and you see this glowing green UIF protein everywhere. But it is especially concentrated and glowing brightly inside that nuclear bud we've been talking about.
Then they take a highly focused, intense laser beam and they blast the bud.
Just zap it.
Zap it. This process, photobleaching, essentially burns out the fluorescence of the proteins in that specific targeted area. They permanently turned off the lights in the bud.
Okay, so if the bud was just an open pocket, like an open cave on the wall of the nucleus, then new, unbleached, glowing green UIF proteins that are floating around in the main nucleus should just drift right into the cave.
And the bud would eventually light up green again.
Exactly.
And to test the baseline, they blasted a random area in the middle of the main nucleus. The fluorescence in that spot recovered almost immediately.
Because it's open.
Right, the proteins were highly mobile, drifting back in to fill the dark spot in just 36 seconds. But when they blasted the nuclear bud with the laser, nothing happened. The fluorescence did not recover at all.
It just stayed dark.
It stayed completely dark. And this raises an important question that proves a key structural reality.
Which is?
The UIF protein and its massive mRNA cargo aren't just sitting in an open depression. They are physically enclosed, locked strictly inside those tiny sealed vesicles within the main bud.
Wow.
They are completely and physically separated from the rest of the main nucleus.
Okay, but hang on. I'm imagining the physical mechanics of this whole process. We know UIF grabs the giant titin transcript, it brings it to the inner wall. The wall bulges in, swallows the cargo, and forms a bubble.
Yeah, we've got that part down.
But how does the bubble actually pinch off and close? If you blow a bubble of bubble gum, you eventually have to close your lips to snap it off. Who cuts the ribbon on this biological package so it can actually leave the dock? Let's do a final quick recap. Titin needs to leave. UIF grabs titin, brings it to the membrane bulges into a bud. But who actually cuts it free?
You're identifying the final, arguably most mechanically difficult step of the process, membrane scission. You have to physically cut the bubble free from the membrane it grew out of.
Which sounds hard.
Bending and cutting a cellular membrane requires a massive amount of energy and highly specialized mechanical force.
So who acts as the molecular scissors?
The researchers looked at a complex of proteins known collectively as ESCRT-III.
ESCRT-III.
In cell biology, the ESCRT-III complex is famous for being the ultimate membrane remodeling machinery. It is the cellular equivalent of heavy-duty automated shears explicitly designed by evolution to bend, constrict, and eventually slice cellular membranes.
So if ESCRT-III is the heavy-duty scissors, what happens if you take the scissors away?
Absolute chaos at the loading dock.
I can imagine.
The researchers once again used genetic tools to deplete the ESCRT-III proteins from the muscle cells. When they looked under the microscope this time, they saw an incredible molecular traffic jam. The UIF protein and the massive titin mRNA were still arriving at the nuclear buds, doing their job, but they weren't getting inside. Instead, they were stuck entirely on the surface, trapped on the periphery of the buds.
It's like an automated bagging machine at a grocery store that completely runs out of heat sealing tape.
That's a great analogy.
The groceries are giant titin transcripts keep piling into the plastic bag, which is the membrane. But without the ESCRT-III scissors to actually cut and seal the bag, it just hangs there on the edge of the machine full of groceries going absolutely nowhere.
Without ESCRT-III to physically snip the vesicles free, the internal packages couldn't form. The cargo couldn't be internalized into the bud center.
It just completely stalled.
The entire special life shipping process stalled out, and once again, the giant transcripts began accumulating, unable to reach the factory floor to build the muscle.
That is just an incredibly elegant system. So if you're trying to picture this whole process in your own muscles right now, this paper by Zaganelli and colleagues really solves a fundamental mystery of biological physics that has stumped people for years.
It really does.
For a long time, we just couldn't explain how these massive genetic blueprints physically relocated from the locked blueprint room to the factory floor without destroying the standard revolving doors.
And now we know.
We now know it requires a beautifully coordinated, highly regulated sequence. First, the internuclear membrane physically bulges inward to create a custom dry dock. Second, the selected handler, UIF, guides the giant cruise ship cargo into the pocket while keeping the small stuff out.
Perfect sorting.
And finally, the heavy-duty molecular scissors, ESCRT-III, snip the package free, sealing it into a vesicle for safe transport.
And the implications of this discovery go far beyond just understanding how you build biceps or hamstrings. This fundamentally changes how we view the nucleus itself.
How so?
The textbook model teaches us that the nucleus is a rigid, static vault with fixed, unchangeable doors. This research proves it is actually a highly dynamic, flexible structure capable of blowing massive inward-facing bubbles to accommodate extreme logistical demands.
And why does this matter for the average person's health? Why should you, the listener, care?
Well, for one, understanding this exact step-by-step pathway could be crucial for treating severe muscle-wasting diseases. If this budding pathway is disrupted, say, by a genetic mutation in UIF or ESCRT-III, the giant proteins required for muscle contraction, like titin and nebulin, can't be built correctly.
Which leads to severe myopathies.
Exactly. Furthermore, if we connect this back to the viral hijackers we discussed earlier, pathogens like herpes simplex and Epstein-Barr virus use these exact same host proteins to infect us and spread.
Right, because they steal the dry dock.
They rely on our UIF and our ESCRT ESCRT-III to escape the nucleus. By mapping how the cell uses these tools naturally, we might find highly targeted new ways to block viruses from stealing them without shutting down the cell's normal operations.
We started today by talking about a highly secure factory trying to move an impossible blueprint. It turns out your cells are master engineers, constantly building temporary, secret exits to keep the machinery of life running. But before we wrap up, I want to leave you with a final provocative thought to chew on.
Let's hear it.
This entire paper focused specifically on muscle cells because they are postmitotic, meaning they don't divide once they're fully formed, and because they have incredibly unique demands for massive proteins. But what about the other highly specialized, long-living cells in your body that also don't divide?
Like neurons.
Exactly. Take the hundreds of billions of neurons in your brain, for instance. Neurons are incredibly complex, they live for decades, and they have massive logistical demands of their own to maintain immense networks.
That's very true.
Are there secret, giant genetic packages bubbling out of the nuclei in your brain right now? Could this exact same dynamic, bubble-blowing pathway be carrying the physical blueprints for your memories? Or building complex synapses in ways we haven't even discovered yet?
That is exactly the kind of frontier question this research opens up. Once you know a secret door exists, you start looking for it everywhere.
Think about that the next time you flex a muscle or form a new memory. Until next time, keep diving deep.